Oh, we got one more minute in it. Okay. Vishali, you back home or you
I'm
in
back
the US?
home. No, I'm back home. Okay. Rajesh actually is in Chicago since 2 days for their digestive disease meet. I'm back.
And Sonakshi is now in St. Louis?
She will be moving to St. Louis. She'll be moving in July, yeah.
Oh, in July, okay.
Yeah, June end. Well, time to start, I guess. Okay.
Yeah.
A very good morning, good evening, whatever, good afternoon to all of you, and very welcome our friends who have joined us once again for the 27th webinar month after month. And we are getting very useful inputs from you, and we are trying to modify our webinars. And this is for the first time speakers. We do get a lot of questions, which we are not able to answer all of them in the last section. So we request all the speakers to please keep on checking Q&A box and keep on typing the answers after your talk or for any talk for that matter to make it interactive. As usual, your CME credit hours will be given once you have filled up the evaluation sheet. And with this, now I will pass on to John Campen and Rima Bansal, who are the part of IUSG Education Committee, to take up and take it forward. Welcome once again.
Thank you kindly, Vishali. So we welcome everyone, and this has been a very successful series. Our first speaker today is Dr. Anita Chen, who is an ocular inflammation and immunology specialist, as well as an anterior segment surgeon at the Singapore National Eye Center and a senior consultant. She's a specialist both in ocular immunology and inflammation, and also in pathology. So she's uniquely qualified to talk about her topic today. So thank you for joining us, Anita, and go ahead and take it away, please.
Right. Thank you very much. Okay, so I'd like to thank IUSG as well as Prof. Vishali for inviting me for this webinar. Today I'll be talking about the pathogenetic mechanisms of primary vitreoretinal lymphoma, and here are my declarations of interest. So the pathogenesis of VRL has not been well elucidated fully, but I'll go through in this talk some of the potential theories of how VRL may occur. And in order to understand how the molecular alterations can lead to B-cell lymphoma genesis, I'll bring you through how a normal B-cell matures from the bone marrow to the germinal center. And then following that, we will go through how these malignant B-cells can pass the blood-retinal barrier and what are the ocular factors that allow these B-cells to home into the retina and how it can proliferate in these immune privileged sites via immune evasion. So I'm going to start with just talking about the normal B-cell development and how the precursor B-cells are formed in the bone marrow and how they go through VDJ recombination that allow for antibody diversity, and these naive mature B-cells then enter the peripheral bloodstream where they may encounter specific antigens. And when they do that, they will migrate to the B-cell follicles and the secondary lymphoid organs, and they will form germinal centers. And within these germinal centers, they will undergo some somatic hypermutation and class switch, and certain B cells will then undergo selection and proliferation before they differentiate into memory B cells and plasma cells. I'll go into this a little bit more detail later, but here I want to ask the question, how can this cell go to the eye and form lymphoma? Are they resident B cells that have been recruited in and undergo de novo malignant transformation, or are they already transformed malignant B cells that go to the brain or the eye? So in order to answer that question, let me go through the normal B cell maturation that goes on in the germinal center. So as I mentioned just now, when the naive B cell encounters antigen, they become activated B cells. They will enter the germinal center via the dark zone and then enter to the light zone where they undergo affinity selection. And then the high affinity cells will go back into the dark zone and undergo clonal expansion and somatic hypermutation. And the ones that have the disadvantageous mutations will undergo apoptosis, whereas the ones that have productive mutations will undergo the class switching and differentiation to form the memory B cells as well as the plasma cells. Now, in this process, a limited number of memory B cells can re-enter into the secondary germinal centers for second round of mutations to enhance the affinity. And it is in this part where if you have an aberrant memory B cell that keeps on going in and gaining additional mutations, they can therefore acquire the mutations to make it into a vitreoretinal lymphoma cell. And we know that these vitreoretinal lymphoma cells have high levels of immunoglobulin aberrant somatic hypermutations, as well as other mutations, especially MYD88 and CD79B, that will increase its proliferation by activation of NF-kappa B. So going back to my question, are they from naive B cells with de novo transformation, or are they from malignant B cells that were retransformed and somehow home into the retina? So because there are no germinal central structures in the brain or eye, therefore there's no structure for them to acquire the mutation. So the theory nowadays that most of us believe is that these tumor cells actually come from have already developed an extraocular site and therefore migrate to the retina and the choroid. And in CNS lymphoma studies, they have actually found evidence of B cell clones in the blood and the bone marrow of patients with primary CNS lymphoma to support this theory that there is actually the lymphoma genesis starts extraocularly. So now the next question is, how do these malignant or VRL cells enter the brain through the or the eye through the blood retinal and blood brain barrier? So we know that in the eye is quite unique. We have two barriers, one similar to the blood-brain barrier, which is the vascular endothelial cells of the tight junctions. But we also have outer blood retinal barrier that's formed by the RPE cells. But in order to understand how it gets into the eye, we'll take some lessons from the CNS, where immune cells are known to be recruited in the vessels. They are activated by adhesion molecules, which allow the B cells to undergo rolling along the blood vessels, and together with certain chemokines and cytokines, they aid the lymphocyte migration and allow them to sort of induce integrin and some changes so that it can tether onto the blood vessel walls and then migrate through the blood vessel walls into the perivascular space. So how does this work in the eye? So many studies have been done, and we know that there are adhesion molecules also in the retinal blood vessels. And we also have chemokines that attract B cells within the retina. So studies have shown that we have ICAM and VCAM that are expressed by human retinal endothelial cells, and these may play a role in the leukocyte migration. And in this study performed and this paper published, what they did was they actually used a simulated retinal endothelium in vitrally, and they blocked the ICAM1 and VCAM1, and they found that it could significantly block the or reduce the human B cell migration across this simulated retinal endothelium. And they found that actually ICAM1 was the better target. And then also in the normal retina, they found that the blood vessel actually partially expresses this chemokine CXCL13, and it's in the so that this chemokine may encourage the B cells to be attracted to the blood retinal vessels. Others have also shown that not only is it in the blood vessels, it's also in the RPE. Now, the RPE is a very important part of where the lymphoma cells are. So later on in your other talks, you will hear more about that. But I want to emphasize that these chemokines are also expressed in the RPE, which is also the other outer blood retinal barrier. So in animal studies where they've injected human CNS xenografts, they also found that these tumor cells do migrate to the eyes, and they also showed the ligand that corresponds to the chemokine CXCL13. And this sort of supports the theory that these lymphoma cells can home into the eyes. And they have found some other ligands such as CD44 as well. So just to summarize that and to give you a visual understanding, because this is really not basic ophthalmology, I'll go through that picture that we showed in the CNS, but now I'm going to transpose it to the outer blood retinal barrier with RPE on top and the vessels. And if they're activated by ICAM or VCAM, they undergo rolling. And these lymphoma cells may have the receptor for CXCR5, and plus the chemokines on the RPE as well as endothelia that sort of attract them to the wall to tether and therefore migrate to the sub-RPE space. And I want you to remember this because later when they go through pathology, you will know that the tumor cell likes to sit in the sub-RPE space. So that may be one mechanism of how the tumor cells, after it goes, the mutational changes, gets hones into the retina and deposits there. So now the next question is, how does it evade the immune system and proliferate as a tumor? So we know that the eye is an immune privileged site. So now I'm going to just briefly talk about the mechanisms of normal ocular immune privilege and how the tumor cell is actually very smart by using this to sort of evade the immune surveillance and proliferate. So we know that the normal mechanisms include the low expression of major histocompatibility complexes. And the vitreous also has a lot of immunosuppressive cytokines, such as fast ligands and transforming growth factor, as well as IL-10. And if there's antigen that is in the eye, they get carried away by the blood vessel to the spleen, where they can activate NKT cells, as well as antigen-specific T regulatory cells that will then produce immunosuppressive cytokines and downregulate the Th1-mediated immune reaction against that specific antigen to keep the eye from having inflammation and having immune tolerance. So how does the B lymphoma cell and VRL take advantage of this? Well, we know that VRL cells produce IL-10, and this also increases the local immune environment for immune tolerance. And we know in patients where we looked at the vitreous cytokines, not only do they have these immunosuppressive cytokines like IL-10, fast ligand, they also secrete the chemoattractant factors like BCA1 and SDF1A, which is the chemokines I mentioned, the older terms for the chemokines I mentioned previously. So they not only do they secrete and enhance the immune suppressive environment, they also have the chemoattractant to attract more cells. We also know that tumor microenvironment plays an important role in helping to suppress anti-tumor activity. And in the eye, we know in vitreoretinal lymphoma, there's always a background of a lot of T cells and macrophages, and they also secrete factors and cytokines that may support the tumor development. So, and finally, we know that the vitreoretinal cells have a very high level of immunoglobulin G mutations, especially a bias towards IgHV4-34, and these can induce antigens. And these VRL antigens, one of which might be galectin-3 protein, because it's recognized by the antibodies using this IgHV4-34 gene. If they are exposed to the APCs and they are carried to the spleen and they activate the antigen-specific T cells, they will further downregulate the Th1-mediated cytokine response and result in proliferation of the tumor. So just to summarize, I've really given you a very brief overview of how the B cell can transform into, with all the mutations that are relevant to VRL, and then how it can home into the eye. So we know that it's most likely from post-germinal centered memory B cells. And what happens is they undergo aberrant recycling into the germinal centers and gain somatic hypermutations, especially in the immunoglobulin genes, as well as other mutations that can activate the NF-kappa B pathways, which allows them to continue to proliferate abnormally. We know that the RPE and the retinal vessels express chemokines and adhesion molecules that can attract malignant B cells. And plus, these malignant B cells have the corresponding ligands to be attracted to these areas. And this is how they can home into the retina. And on top of that, they will undergo rolling, adhesion, and diapediasis with these adhesion molecules to enter into the sub-RPE space or the perivascular space. And to proliferate, they secrete immunosuppressive cytokines and have a tumor microenvironment that will take advantage of the immune privileged nature of the eyes to allow these malignant B cells to evade cytotoxic T cells and proliferate in the eye. So with that, I'll end my talk and hand over to the rest of it.
So thank you, Anita. That was a wonderful overview. I wouldn't call it brief, very knowledgeable and very interesting. So thank you so much for that. So with this, we now move on to the second talk of our session. For this, we have Dr. Deborah Goldstein, and everybody knows her. So she's a professor of ophthalmology and director of Uveitis Services and Uveitis Fellowship in the Department of Ophthalmology in Northwestern University, Feinberg School of Medicine. So Dr. Deborah will be talking on the clinical presentations and differential diagnosis.
Thank you so much. Let's see if I can pull up my screen. Here we go. So we're going to switch gears. I am trying to get into, there we go. That's not what I want though. I'm going to switch gears now. We just heard a lovely talk about the pathogenesis. So I'm going to talk a little bit just about the clinical presentation, and I'll try not to step on David Saraf's toes and try not to show too many images, but we have to show some images. So this is really just going to be clinical cases. So my first case is a 44-year-old male who's had floaters in both eyes for more than 2 years. And I want you to pay attention to just how young this guy is. He's been diagnosed elsewhere with idiopathic vitritis and had therapeutic pars plana vitrectomies with the specimen thrown in the garbage. When I see him, he has 2+ large vitreous cells and an opacified vitreous skirt. And we finally convinced him to have another vitrectomy, and the diagnosis was consistent with PVRL. And what I'm going to do now is just show you some of the pictures of the various presentations that he had in the retina through his course. So here you can see, and I don't know if you can you see my pointer or no? No.
Yes, Deborah, we can
Okay.
see. Yeah.
Good, thank you. So here you can see that there's some superficial retinal infiltrates. So it's not the classic subretinal or sub-RPE lesions that we see, but this is actually very common. Outer retinal, inner retinal, pre-retinal, all of these things are possible. So this is one of his presentations. Here's another recurrence that he had, and this is what we see or expect more typically, this subretinal or sub-RPE collection of cells. And here you can see on the OCT that this particular collection is sub-RPE. This is another recurrence, and this actually shows you the multiple different locations that can be involved. There's actually the residual vitreous skirt, which is filled with large white cells. And in this picture, you can see he has superficial retinal, deep retinal, subretinal involvement, all of these different infiltrates. And while in most of these cases, these resolved with treatment, I will tell you that these can also appear to migrate or change even without treatment. So here you can see that there is outer retinal and subretinal involvement of these lesions. Here you can actually see one of the vertical hyperreflective infiltrates. This is another type of presentation that we could see, which is very superficial or on the retina. And this is what these lesions look like, these white infiltrates that appear to be sitting on the retina or in the superficial retina. And here imaging is very helpful, but I won't steal David Saraf's thunder. Here is a picture of one of the superficial retinal infiltrates on your left, and on the right is the same area after intravitreal methotrexate. And you can see here superficial retinal infiltrates. It kind of, for anybody who's watched The Simpsons, this looks like Bart Simpson's hair, and this goes away with treatment. So just to show you from this patient, A, he was in his very early 40s when this started, and B, he did show all kinds of different retinal infiltrates, every layer of the retina. And the third is to make a plug that there is no such thing as a therapeutic vitrectomy in uveitis where you don't send the specimen to pathology. Our next case is a 61-year-old male whom I just saw a few months ago, and his complaint is of 3 months of floaters and photopsias, that shouldn't say photophobia. And he's referred from an outside retina specialist. He's been on topical corticosteroids. He denies medical problems, but does not have a regular physician. So you can see on this picture, well, first, he has 20/20 vision in both eyes, does not have much going on in the anterior chamber, but has 3+ large, very chalky white vitreous cells. And that's really characteristic of PVRL or VRL, is these chalky white cells. And all you can see on these photos is really just these drusenoid changes. So somebody with vitreous cells and drusen, just think about, are these really drusen? My current fellow, who will be joining me in practice in a few months, did this slide for me. I have no idea how to do this, but he's showing you the enlarged areas of these multiple drusenoid deposits. And here you can see on fundus autofluorescence, all of these deposits are uniformly hyperautofluorescent. And this could be a clue differentiating these from drusen, which can be hyperautofluorescent, hypoautofluorescent, or isofluorescent. The PVRL ones tend to be all hyperautofluorescent. And again, sorry, David, we have some OCT images to show you that these deposits are subretinal. And in this case, this was in his right eye only. His left eye was completely normal. So his workup for syphilis, sarcoid, and tuberculosis were negative. And we did an AC tap for my MYD88 mutation, which was positive. He had a brain MRI that was read as normal. And unfortunately, the gentleman refused diagnostic vitrectomy and refused care. And he subsequently cycled through multiple different institutions looking for a different diagnosis than VRL. And 2 months later at a different institution, he had a repeat MRI brain, at this point showing an ill-defined enhancing lesion in the right frontal lobe, consistent with CNS lymphoma, probably to go along with what we believe is PVRL. The last case I'm going to show is a 75-year-old woman with decreased vision in her right eye. She has not decreased vision. She has vitreous cell and haze only in her right eye. This is a picture that I took to show you what I'm talking about with the vitreous cells. These are very large, chalky white vitreous cells. These look different from the normal vitreous cells that we see in non-malignant uveitis. These are very large and very chalky white. So here's her fundus picture, and you can see that there's an area here of what looks like viral retinitis. And I chose this case to show you because while this isn't typical, PVRL can present looking like viral retinitis. I will tell you that at this point, we already knew that she had biopsy-proven vitreoretinal lymphoma. So the differential for this, as I said, viral retinitis, we can think of zoster being most likely in her age group, herpes simplex, CMV, also toxoplasmosis if she's immunosuppressed. But again, she does have biopsy-proven VRL. And here's an OCT of the lesion, and you could see dramatic retinal thickening, but you also see breaks through the RPE with these lesions coming through the RPE. So we can see these jelly bean-like lesions in sarcoidosis actually, but we don't tend to see this big, dense, huge, full thickness retinal infiltrate. So why is this not herpetic disease when we look at this OCT? And one of the clues I'm going to give you is this is after treatment, and you can see that there is relative preservation of the retinal layers. When we have full thickness retinal necrosis with toxoplasmosis or herpes, we tend to get marked atrophy afterwards rather than preservation of the retinal layers. And here you can see there's another episode where she has a recurrence. And again, you could see this white retinal infiltrate that absolutely could be confused with viral retinitis. And again, on the OCT, you see this massive retinal thickening, which is actually much thicker than we get with retinal necrosis from viral infection. And here is after treatment. Here, the retina doesn't look great, but you still do see preservation of some retinal layers. So again, this is the first image that I showed you, the full thickness retinal necrosis or full thickness retinal infiltrate, but with resolution, preservation of some of the layers. And this is in contrast to viral retinitis. This is a patient of mine with herpes zoster, acute retinal necrosis. And you could see after the resolution of the retinal infiltrate, we've got almost complete loss of the retinal layers. Here's another patient with an infectious retinitis that is in the differential. You can see that there is retinal infiltration. There's some subretinal fluid. Here there's choroidal inflammation underneath it. And with resolution, we have focal loss of the retinal layers. And actually, one of our current fellows published a lovely paper on features that can be used to distinguish retinitis-like lesions in VRL from infectious retinitis. This is Alessandro Marchesi from Italy. And we can see that the retinal infiltrate is much thicker with disorganization of all of the involved layers. We can see hyperreflective infiltrates in the subretinal and sub-RPE space, and very importantly, partial restoration of the retinal layers after therapy. I'm going to end with the concept that I didn't learn when I was a resident and fellow, but have seen dramatically over the years that we can get very significant anterior segment involvement in patients with vitreoretinal lymphoma. So this is a 62-year-old patient with biopsy-proven vitreoretinal lymphoma, and she presents a few years later with these very large gray-white chalky carotid precipitates. And I did an anterior chamber tap, which showed enlarged atypical lymphoid cells, condensed chromatin, scant cytoplasm that was consistent with lymphoma. So there was one report in 2000 of anterior segment inflammation in approximately 1/4 of eyes with VRL at presentation. And we have a series from Italy and from Chicago looking at a retrospective analysis of 108 eyes of 55 VRL patients. And anterior segment involvement was seen in 50% of eyes at least one point. So this isn't necessarily at presentation, as the woman I just showed you, but at some point, the development of AC inflammation. KP were the most common, and the KP were present in almost 50% of patients. And most commonly, these were creamy, large white KP, but in some cases, these were pigmented. And we did see AC cells as well in almost half the eyes. So I urge you, if you see a patient with chalky white cells in the vitreous and you're suspecting VRL, don't discount vitreoretinal lymphoma because of the presence of KP. So I'm going to summarize here, and for once in my entire life, I'm going to end before my time rather than after. I tried very hard, Vishali. Although VRL is more common in older patients, I have a number of patients in their 40s, and in all cases, the diagnosis was missed because the diagnosis was discarded based on the patient's age. So don't discard the diagnosis and don't discard a vitreous specimen. You can certainly see VRL in younger patients. You can have anterior chamber cells and KP. The vitreous cells, typically large, white, and chalky. I didn't mention, but the CME can occur, but it's very rare. And the infiltrates that we see can be anywhere in the retina. They can be pre-retinal, they can be intraretinal, very commonly outer retinal, and then what we classically think of, subretinal or sub-RPE. Thank you.
Thank you so much, Debbie. That was great. I always love all of your cases. They're so instructive, and the way that you narrate it really helps to remember these things. So thank you very much. Yeah.
Thanks.
So our next speaker is David Seraph. So David is a UCLA Jules Stein faculty member who is seeing a broad spectrum of retinal diseases overlapping into ocular inflammation. And he's had an illustrious career in clinical research and identifying of novel syndromes. And so he's the perfect person to teach us about imaging characteristics of vitreoretinal lymphoma. Thank you very much for joining us, David.
My pleasure. Can you see my slides?
Yes.
Yes, David, all good.
All right, that's awesome. Well, thank you very much for the opportunity to speak. It's great to see everyone, and I'm very happy to be part of this very instructive symposium. Vishali asked me to speak of the imaging characteristics of primary vitreoretinal lymphoma. Here are my disclosures, and I'm more than happy to focus on that topic and to highlight a paper that was written by Francesco Picchi that we published recently in the American Journal of Ophthalmology. That will be the centerpiece of this talk. So as we all know, ocular lymphoma can present in two major ways. There's choroidal lymphoma, in which the infiltrates typically are deeper in the choroid without retinal involvement, and often in the category of more benign forms of lymphoma like maltoma. And then there's the more malignant form of ocular lymphoma that we refer to as primary vitreoretinal lymphoma or vitreoretinal lymphoma that has the high association with CNS disease. And this talk will focus, of course, on primary vitreoretinal lymphoma like the other talks, and we'll focus on the imaging characteristics because these can be so important because the disease can be so challenging to identify and diagnose and can masquerade as other conditions. As Deborah Goldstein has just shown, these can masquerade as AMD or uveitis. And so imaging, especially advanced retinal imaging like OCT, can be critical for the early detection and can guide indication for tissue diagnosis. And this one study that we embarked upon was specifically, the objective was to determine the OCT characteristics of this disease to make diagnosis more easy and to facilitate diagnosis. So the objective of this study was to analyze the spectrum of OCT findings in patients specifically with biopsy-proven vitreoretinal lymphoma and correlate these with some of the clinical parameters, including the systemic parameters of this disease. And as I mentioned, this study was led by Francesco Picchi. Some of our panel members and speakers were co-authors like Deborah on this paper. And it was published just a couple of years ago in the American Journal of Ophthalmology. This was a retrospective observational study. We did a multi-center chart review from 13 centers, including ocular oncology clinics worldwide, who saw patients over an 11-year period. And we required every patient in this study to have biopsy-proven vitreoretinal lymphoma, which is a very unique aspect, I think, of this investigation. All patients were required to have high quality spectral domain OCT and to be treatment naive at the time of baseline imaging. And Dr. Picchi and I reviewed all of the OCT images. We also looked at FAF, which we'll briefly discuss, fundus autofluorescence, and fluorescein angiography. In total, we were able to recruit 115 patients for review, which is, I think, the largest study now looking at imaging, OCT imaging characteristics of primary vitreoretinal lymphoma. There was a slight predominance of women in our cohort, and most of the cases were bilateral with a mean visual acuity of 20/30. At baseline, a third of the patients had isolated ocular disease, and 50%, not surprisingly, had associated CNS involvement. What's very interesting is that 10% of the patients had non-CNS systemic involvement, and another 10% had both CNS and non-systemic CNS involvement. So the teaching has and the bias has always been that primary vitreoretinal lymphoma does not have non-CNS systemic involvement, but according to our study, at least, you can have non-CNS systemic involvement in a significant 20% of cases. What was very interesting about this study is that the OCT, which is a depth resolved imaging modality, permitted us to look at the breakdown in the localization of the infiltrates. And as Deborah already mentioned, you can see these infiltrates in the retina, in the subretinal compartment, and in the choroidal compartment. But in our study, by and large, most of the infiltrates had sub-RPE involvement, and this was by far the most common location of the infiltrates that we saw with OCT. Subretinal involvement was also very common in almost 50% of cases. To see intraretinal involvement and a couple of those cases that Deborah just showed with full thickness intraretinal involvement, that was a more unusual presentation in our international multi-center consortium. And here you can see the breakdown using this Venn diagram. By far the most common presentation with OCT were infiltrates in the sub-RPE compartment, and these were often isolated to that location. And then also we could see lots of cases, the majority of cases either had sub-RPE or combined sub-RPE and subretinal involvement. To have isolated intraretinal or combined intraretinal involvement was certainly a significant presentation, but it was by and large a more uncommon one. And here are some of the OCT illustrations that Francesco Picchi was able to put together that really, I think, illustrate beautifully the characteristic OCT findings of this disease. And as already mentioned again by Deborah, you could see these drusenoid lesions, and it wasn't unusual to see large drusenoid RPE detachments masquerading as AMD. The second most common presentation were these hyperreflective subretinal deposits, which often had this kind of cuff-like appearance on the OCT, and sometimes you could see these bands of hyperreflectivity in the subretinal department. Now, intraretinal involvement was also possible, although it was much more uncommon, and you can see these full thickness infiltrates that Deborah also showed, and often they had this kind of vertical band-like phenotype, which is almost diagnostic of vitreoretinal lymphoma. Some of our patients, 13% and 11%, had intraretinal and subretinal fluid respectively, and the subretinal fluid was often associated with these large drusenoid RPE detachments, and this could relate possibly to RPE pump failure when you have significant displacement of the RPE from the underlying choroid. So fluid was not an unusual accompaniment of these subretinal and sub-RPE infiltrates in patients with vitreoretinal lymphoma. We also looked at this phenotype referred to as cloudy vitelliform lesions, originally described by the Freund-Januzzi group, and 10% of our patients presented with this cloudy vitelliform lesion, this vitelliform-like lesion, hyperreflective vitelliform-like lesion in the central compartment at the subretinal location. And this can be a transient finding of primary vitreoretinal lymphoma, but is very characteristic, and I would say even diagnostic of the condition warranting vitreoretinal biopsy. I mentioned that we did fundus autofluorescence as well in this study, and 55% of our cases had abnormal fundus autofluorescent findings. The most common presentation on autofluorescence was this leopard spot appearance, where you saw both hyper and hypoautofluorescent lesions with wide field fundus autofluorescence. But we also saw patients with just hyperautofluorescent lesions kind of masquerading as mudes. And in younger patients with vitreoretinal lymphoma, it's important to understand, especially when the presentation is initially unilateral, that patients can present with can be confused and masquerade as mudes with these ephemeral or transient hyperautofluorescent lesions with fundus autofluorescence. But when you do the OCT and you see these characteristic cuff-like subretinal hyperreflective lesions, that should lead you to the diagnosis of vitreoretinal lymphoma. and to the necessity to obtain a vitreoretinal biopsy. And here is actually a case presentation that I'll end this lecture with. A patient who presented with what was thought was initially MUDES with these hyperautofluorescent lesions. But if you look carefully, you'll see some hypoautofluorescent lesions as well. And that can be a clue that this is not a MUDES and it's actually a MUDES masquerade. This patient went on to develop more of a leopard spot pattern on the fundus autofluorescence with wide field imaging, and then developed these hyperautofluorescent lesions in the left eye, another sign that this is not MUDES and that you're dealing with a more malignant consideration here. And on FA, you can see the characteristic leopard spot pattern, which was also a characteristic phenotype we saw in our AJO study. This patient also had a central cloudy hyperreflective vitelliform lesion that was transient and resolved. And as I mentioned in our study, we saw that in 10% of the cases. And then went on to develop the characteristic cuff-like and band-like areas of subretinal hyperreflective infiltration, as you can see the arrow showing the progression of disease. And then ultimately developed the characteristic drusenoid RPE detachments with the vitreal infiltration. And then we see that the patient developed this creamy infiltrate, which is almost diagnostic. The patient went on to vitreoretinal biopsy of this lesion, and sure enough showed large atypical lymphocytes that were positive for CD20. So in summary, RPE infiltration is the most common OCT manifestation of vitreoretinal lymphoma. Subretinal combination subretinal lesions are also common, and look for those hyperreflective cuffs and hyperreflective band-like lesions. Intraretinal lesions can also be a form of vitreoretinal lymphoma with worse visual acuity, but these are the rarest form of lesions. Large RPE detachments can also present with fluid and can masquerade as AMD. In young patients with autofluorescence, this condition can masquerade as MUDES. The fact that these lesions are most common in the RPE and subretinal compartment would indicate perhaps actually that vitreoretinal lymphoma is originating from the choroid, but that still requires further investigation. Thanks very much for the opportunity to speak again. Appreciate it.
Thanks for getting up so early to speak to us.
Oh, my pleasure. Not so bad, actually, 5 o'clock.
So thank you, Professor Saraf. And we now move on to our next talk on this. And it's a pleasure to introduce Professor Sarah Copeland. She's a professor and a senior consultant histopathology at the histopathologist at the Liverpool University in UK. And she leads the world-renowned Liverpool Ocular Oncology Research Group, and we're known for her expertise in ophthalmic pathology. She's the best to teach us on the diagnostics from intraocular fluid.
Thank you very much. I'll share my screen now. Can you see that?
Yes.
Okay, I'll put it on slideshow now. Okay, thank you very much for the, sorry. Thank you very much for the invitation.
Yeah.
Okay. Thank you very much for the invitation to speak. I'll be going through some of the diagnostic work, and I think I might be repeating some of the things that have already been said, but I'll skip through the slides quickly. So, as you probably all know, lymphomas can be essentially divided into Hodgkin and non-Hodgkin lymphomas, and the non-Hodgkin lymphomas can be divided according to B, T, and NK subtypes. They can also be divided according to where they arise, whether they occur in lymph nodes or extranodal sites. And obviously, the eye and the ocular nexa are extranodal sites. So when we, as pathologists, make diagnosis of vitreoretinal lymphomas, we use the WHO lymphoma classification, which takes into account various features, including the clinical aspects, the histo or cytomorphology, the immunophenotype, and also the molecular alterations. over the years, there have been various iterations of the WHO lymphoma classification. And last year, there was an update of this lymphoma classification, which took into account more advances in molecular pathology, including the genomics, the transcriptomics, proteomics, and some metabolomics. I was involved in the editorial board of the WHO lymphoma classification, and this the revision of this volume, tumor volume, involved a large number of people and involved also a large number of countries. Because it was the COVID period, a lot of the interactions had to take place via Zoom, as we're doing today. However, despite this, a huge amount of work was achieved. These were the timelines for the actual publishing of the WHO 5th edition. Unfortunately, things have been slightly delayed, and so it's still not out in print. However, hopefully that will take place in summer this year. However, there were 2 publications mid last year which outline the lymphoid neoplasms and also the myeloid histiocytic dendritic neoplasms and the classification thereof. So what does this have to do with ocular lymphomas? As you all know, the ocular lymphomas, this broad umbrella, includes the ocular nexal lymphomas and the intraocular lymphomas. Won't have time today to speak about the ocular nexal lymphomas, and certainly we won't have time also to speak about all of the different intraocular lymphomas. However, they have already been mentioned by a couple of the speakers, and primary choroidal lymphomas are low-grade B-cell lymphomas, which are to be distinguished distinctly from the vitreoretinal lymphomas, which are high-grade B-cell lymphomas, which can have manifestations in the central nervous system. When it comes to the new WHO lymphoma classification, these vitreoretinal lymphomas are still considered to be diffuse large cell B-cell lymphomas, as the vast majority of them are. But it should be kept in mind that there can also be primary T-cell lymphomas occurring in the eye. But the primary diffuse large cell B-cell lymphomas within the eye have been sort of not subsumed, but included within a larger group called DLBLs of an immune privileged site. And these belonging to this large group are those occurring within the testis and also obviously occurring within the brain. So there are advantages, I should say that there are advantages of having such an umbrella term, in that by comparing what occurs in the vitreous versus the brain versus the testis, then we can better understand the biology and the pathogenesis of these tumors, which was first outlined by the first speaker, Anita, in this symposium. So I'm not going to go through this. This has already been covered by the clinicians, who have a lot better idea of the clinical features of vitreoretinal lymphoma than I do. But I guess what I'd like to highlight are the ways we make the diagnosis. So obviously close interaction with the clinicians. Typically, a vitrectomy is performed. Sometimes you can have subretinal aspirates, and you can have also chorioretinal biopsies, something that I'll return to. And in very advanced disease, you can have an enucleation, but this is obviously the rarer scenario. So what we do is that we receive the vitrectomy, and these can be the diluted or the undiluted samples. We prepare cytospins, and we stain them with a standard stain called May-Grunewald-Ginza. And then we perform the immunophenotyping using immunocytology. And so we use CD20, which is a marker for B cells. We obviously do the CD3 to compare that. And then we also use CD68 to make sure that we're not looking at activated macrophages. We also perform Ki67. We typically then put aside the sample, the remaining part of the sample, and from there we extract the DNA to do the molecular analyses, which I'll come on to. This is an example of a chorioretinal biopsy, and you can see the tumor cells and the disruption of the retinal pigment epithelium, which was referred to in the OCT finders by the last speaker. So some people do the immunophenotyping using flow cytometry. This is all very much dependent on the lab and the lab's experience. And they use various gating mechanisms to highlight the B cells, the neoplastic B cells compared to the T cells. Others use ELISA, and they look at the interleukin 6 and 10 ratio. Some also include interferon gamma. We usually reserve some of the specimen, as I mentioned, to do the clonality analysis, where we look at the rearrangements of the immunoglobulin heavy and light chains. And we also try and reserve some of the sample to do the MYD88 mutational analysis, which is present in approximately 70% of the vitreoretinal lymphomas. What is happening more and more in cancer diagnostics is looking at liquid biopsies, and obviously when we're referring to the aqueous and the vitreous, these are liquid biopsies, but what I'm referring to here is people are looking at within the blood to look at circulating tumor cells and also circulating free DNA, and also things called exosomes, which may contain microRNAs. And so I'll move on to that with respect to the eye. So there are some centers which perhaps more in a research arena using differing techniques, and this is ultrasensitive droplet digital PCR, where they've looked at the aqueous only and have been able to demonstrate in the cases of frank vitreoretinal lymphoma that MYD88 mutations could be demonstrated without any accompanying cytological analysis. There are other centers, such as in the US, where the metagenomic deep sequencing of the aqueous has also been undertaken, looking specifically for the MYD88 mutation. Then there are others who've designed next generation sequencing panels, and where they're looking at cell-free DNA and also the tumor cells within the specimens, and have targeted against MYD88 and these two further genes. Over time, and this was a study that I was involved in, an improved next generation sequencing panel was designed by the group in Tübingen in Germany, where they have looked at a wider panel of genetic mutations and also copy number variations to help make a definitive diagnosis of vitreoretinal lymphoma versus a chronic vitritis. And then again, this is a very recent paper, came out last year, looking at cell-free DNA using, again, a broader next generation sequencing panel. So I'm just going to, and something that was published a while ago, but hasn't been validated as yet by another center. It's looking at microRNAs, which are released from so-called exosomes, which are secreted by the tumor cells. And this is essentially looking at the supernatant of the vitrectomy rather than the cellular component of it. And so this does require validation, but they suggest that using this constellation of the microRNAs, you can differentiate a vitreoretinal lymphoma from a vitritis. This is just an example of a chorioretinal biopsy. These are performed in some centers where perhaps a vitrectomy hasn't provided with a definitive diagnosis. The advantages of a chorioretinal biopsy is obviously for the pathologist. We can play around with more antibodies, and we can also do a range of different tests. And as Anita mentioned in her lecture, the majority of the vitreoretinal lymphomas are diffuse large cell B-cell lymphomas of so-called ABC subtype, and they express this MUM1 and BCL6, and a normal B-cell does not co-express these two molecules. It's either one or the other. And so this is a demonstration that these are neoplastic B cells. And yes, the very high Ki67 growth fraction, virtually 95% of these cells are proliferating if they're still viable. Whereas if you were to compare this with a chororetinal lymphoma, a choroidal lymphoma, the proliferation rate would be exceedingly low, maximum, say, 5%. Just want to highlight this paper. This was the one that I mentioned before, where there was a multi-center collaborative study looking at vitreoretinal lymphomas, those occurring primarily within the eye and those that occurred secondarily, for example, as a result of a systemic involvement of a systemic non-Hodgkin lymphoma. The key features of the primary vitreoretinal lymphomas are these mutations in MYD88, CD79B, and PIM1. Fewer mutations in these two genes, but they also demonstrated this copy number variation involving chromosome 9. So this is sort of like a heat map. I don't unfortunately have time to go into great detail about it, but they highlighted these key top genes which were involved. And this was then validated by a study conducted by this group, and the results came out in July last year. Again, sort of highlighting these top hits if you wanted to design a next generation sequencing panel for vitreoretinal lymphoma. Just want to say that this does look rather complicated, but this is a new sort of molecular subtyping system of diffuse large cell B-cell lymphomas according to the cell of origin, or COO. And when it comes to the vitreoretinal lymphomas, the CNS lymphomas and the testicular lymphomas, they all have this characteristic sort of mutational profile, which has been called C5 or MCD or MYD88 or CD79 beta mutated. And this is the paper which was put forward by Björn Chapuis, who's now located in Berlin. This is the umbrella group of diffuse large cell B-cell lymphomas of immune-privileged sites. And they have some common features, but obviously not consistently the same. However, as I mentioned right at the very beginning, it is probably of value to for our better understanding of the vitreoretinal lymphomas to work alongside scientists who are examining the CNS and the testicular lymphomas, just so that we can better understand this disease and come up with better treatments. So this is a summary. The clinical features I won't go into. The vast majority of B-cell lymphomas either coming up with CD20 or CD79A, and they have a huge number of mutations, which I think Anita mentioned in her talk. These are somatic mutations which occur during the so-called germinal center reaction, and they are highly likely to be these neoplastic cells probably have arisen from so-called post-germinal center B cells, and probably of ABC subtype, possibly of the C5MCD subtype, again requiring further validation. We need to differentiate these tumors from microorganisms such as toxoplasmosis. Deborah mentioned this in her talk. This was a case that I saw where I sent a vitrectomy sample where the toxoplasmosis cysts were obviously present in the sample, and obviously also in patients who may be immunosuppressed, that they have candida, you differentiate them, and also aspergillosis. A plea from a pathologist is when you're sending in intraocular samples, don't put them in glutaraldehyde. The years of electron microscopy for the diagnosis of tumors within the eye is really gone. If you are sending a tissue biopsy, like a chorioretinoid biopsy, then please put that in formalin, buffered formalin. However, if you're sending in fluid samples like vitrectomies, then send them in a soft fixative such as Cytolite or hope fixation, This does enable a quicker turnaround of the samples and the creation of these cytospins, which I referred to at the very beginning of my talk. If you suspect a microorganism, then send the sample in fresh. But most importantly, speak to your pathologist. Let us know that when you're sending in the sample, so that we are prepared for it, we can look out for it, and we can work it up most efficiently and without wastage of these precious, the precious material. Just to highlight, this is a very old paper of Berto D'Amato and myself. However, it does provide the background of understanding enterocular lymphomas, particularly the vitreoretinal lymphomas and the primary choroidal lymphomas versus systemic non-Hodgkin lymphomas, which secondarily infiltrate the eye. So just to summarize, I'm a pretty perhaps conventional pathologist. I still would like to see cytology in the of a specimen before making a diagnosis and combining that with any of the molecular techniques. We are living through a very exciting period with the advancements of a whole range of different molecular tests, which are becoming cheaper and cheaper, which is great, so that we can provide them for our patients. By understanding the biology of the tumor cells, then we can come up with targeted therapy, which are being tested out in various clinical trials. And it's really through concerted symposia like this and concerted clinical trials that we will be able to improve the outcome of these patients. This is the sun rising over Liverpool, over the Mersey River. There was the coronation today, as you probably all know, but perhaps even more exciting event is this time next week is the Eurovision contest, which will be taking place in Liverpool. The King and Camilla were here last week and pressed a button to open the Eurovision to get it on its way, a bit like him getting on it his way or their way during the coronation. So thank you very much for your attention, and I'm happy to take questions now, or if people would like to send me questions via email. Thank you very much.
Well, thank you very much. That is really informative. I think we're going to try to do the questions at the end, which is about another 30 minutes, if you don't mind. So let's go ahead then to our next speaker. I think Professor Jose Pulido is very well known to many of us. After an extremely illustrious career at Mayo, he's moved to Wills Eye Hospital, where he's continuing that illustrious career as the Larry Donoso Endowed Chair and the Director of an endowed translational medicine laboratory and research center. He's professor in the Department of Ophthalmology of the Sydney Kimmel Medical College at Thomas Jefferson University, which is Wills is the primary eye location for that. And he seems to know nearly everything clinically. And so I'm looking forward to his talk about clinical implications of histopathological findings. Jose?
His is recorded talk, it will be played. So can we play that talk?
Sure.
Jose Pulido, I'm sorry that I can't be with you, but there's a conflict because the timing had been told to me incorrectly. I was told it was 7 o'clock Eastern time. It was actually 8 o'clock Eastern time because of the spring forward confusion. No matter, I will give you my talk. I myself don't like pre-recorded talks, so I'll try to do this talk quickly because being on the other side, I know how pre-recorded talks can be a little dull. So the question is tissue the issue anymore? I have no financial disclosures to talk about here, and I'm going to give you the conclusions for the start. And that is, if you're going to do a vitrectomy, then do an extensive vitrectomy. and send for MIGHTY88 PCR and histology. Forget flow. Secondly, if you're going to do it, it's actually helpful because it debulks the amount of lymphoma cells since they have a huge Ki67, which is a sign of proliferation. That's actually helping the situation. So this is based on 2 of our papers diagnosing the vitreoretinal lymphomas and histologic findings in vitreoretinal lymphoma, learning from enucleation specimens. And I think it's important to read these papers, especially the second. And we'll also be mentioning the following papers. So the first paper mentioned shows that flow cytometry is actually pretty bad because it has a very poor coefficient of variation, and cytology actually has a very poor coefficient of variation. In other words, if you do it and then do it again, there's a great chance of differences. Well, as you can see, the coefficient of variation for MIGHTY88 is pretty low. So MIGHTY88, even though it has about a 70 to 80% chance of being positive, MIGHTY88 PCR, remember that MIGHTY88 L265P is mutated in about 75 to 80% of the cases of B-cell lymphoma. 5% of vitreoretinal lymphomas are T-cell lymphomas, and they have inflammation, very different beasts. B-cell lymphomas rarely have inflammation, about 15% of the time, and are can have this constitutively activated MIGHTY88. And MIGHTY88 with this L265P mutation is turned on all the time, and so the cells are on constantly. The B cells are on constantly. So the first order of business before considering vitrectomy, etc., is, has this patient been staged? And initial staging involves whole body PET and an MRI of the brain. And if the MRI is positive, then the CNS biopsy is sufficient. You don't have to do the vitrectomy in the eye unless you want to do it for the reasons we'll talk about shortly. But tissue, when there's CNS involvement or testicular involvement, is an issue. So what if there aren't any MRI findings? Then cell-free DNA looking for the MIGHTY88 L265P is reasonable, but if you don't have cell-free DNA testing, then a vitrectomy is very good. And if you're going to do it, go the whole way and get a copious sample. The way I think it's reasonable to do it, put a syringe on a vitrector, have the infusion turned off. As far as the cut rate and the gauge, the data says no difference, whether you use high cut rate or low cut rate, and small gauge or large gauge. But I like to use a cut rate of about 350 and a gauge of about 25, no smaller than 25 gauge. I find 27 gauge doesn't work quite as well for the vitrectomy for biopsies. But again, the data says there isn't a difference. What I like to do is to turn the infusion on and off as I go to different parts of the vitreous cavity and go to form vitreous. So I start initially in the center, start the vitrector, then after I'm cutting, then I tell my assistant to aspirate. Never aspirate when you're not cutting. Then when the eye starts to become slightly hypotenuse, I tell my assistant to stop aspirating, and then I stop cutting. That's the order. I tell them to turn the infusion on. The eye reforms. Go to a different part of the vitreous cavity and do the same thing until you have about 10 cc's of semi-formed vitreous. After that, you hook up the vitrector to the automatic cassette and you do a very complete vitrectomy. You send both the semi-formed vitreous and the cassette for a cell block, and that's about it. So why a wide vitrectomy? Well, there's a case report in the literature that just doing the vitrectomy helps to keep the lymphoma cells from recurring. And the question is whether you remove the scaffold and whether that helps, and whether increasing the oxygen levels breaks down the blood-retinal barrier. Nobody knows, but you do debulk the vitreous. As I said, they have a very high Ki-67, and so debulking helps in decreasing the chances of recurrence. And when you do that, it's very important to remove the anterior hyaloid as best as possible. Now, that's a lot harder to do in a phakic eye, and I understand that, but it is important to look at this carefully. And here's a case where the patient had lymphoma cells growing in the anterior hyaloid, and we repeated a vitrectomy and removed and debulked that anterior hyaloid Berger space. Now, what about cases like below? Well, obviously, those are easy, and there's plenty of cells to be had, and this is where you do a wide vitrectomy, and you can see the cells attaching to the anterior hyaloid face. But what about the subretinal cases like this? This is not one where I would consider a vitrectomy. There aren't that many cells. The vitreoretinal lymphoma cells are mainly underneath the retinal pigment epithelium, and there's not that many of them. On the other hand, this case has sufficient, and it is reasonable to consider doing a vitrectomy. And if there are enough cells, stopping there, but if there aren't enough cells, then attacking the cells in the sub-RPE space. And to do that, you just need to use the same technique that we described before, using the vitrector and the syringe and taking little bits. You don't have to do a chorioretinal biopsy, but I'll tell you why a chorioretinal biopsy is silly, actually. Here is from the paper on histology that we had, and again, I really urge you to read this paper. Here you can see a case of vitreoretinal lymphoma. All the cells are anterior to Bruch's membrane. All the CD20 cells are anterior to Bruch's membrane, and below Bruch's membrane are only CD3 cells. So if you do a chorioretinal biopsy, you're putting the CD20 cells and the CD3 cells all kind of together, and there's no need, and you're going to just obfuscate the whole situation. Here's another case of the same thing. Anterior to Bruch's membrane, CD20 positive, rare CD20 cells below Bruch's membrane, and the reverse, plenty of CD3 inflammatory cells below Bruch's membrane and none anterior. Don't do a chorioretinal biopsy. It's worthless. Just take a chunk of the retina, and you can laser around it if you want to. So those are the important things to consider in this in these situations. And I wish again that I could be with you. Remember that vitreoretinal lymphoma is like the evil Shredder in Teenage Mutant Ninja Turtles, and you have to give them no slack. Get that vitreoretinal lymphoma. Thank you.
So thank you, Professor Pulido. That was very kind of you to have given us the recorded talk. And so with this, we now move on to the last topic of the this webinar, and that's on management of vitreoretinal lymphoma. And for this, it's a pleasure to have Dr. Anita Agarwal. We all know her for her vast experience, for almost 4 decades of experience in ophthalmology, and not only the long experience, but vast experience in medical retina, surgical retina, inherited retinal diseases, and uveitis. So she sees all these patients and has extensive work on it. So Dr. Anita, for your talk on management, please.
We can't hear you, Anita.
Mute myself.
Now we can.
Start video.
There we go.
All right. Okay, here we go. Sorry about that. Can you see my screen?
Yes. It's
You
not
can?
on slideshow though. Yes, we can.
Okay, yeah. Thank you, everybody, for having me participate in this. And you know, I guess all of us are far more, as ophthalmologists and retina specialists, more involved in clinical diagnosis and things like that rather than the management. Because as you know, the management is a sort of a shared management with oncologists. But I'll try to do my best as to what I can, as we as ophthalmologists and retina specialists or ocular oncologists can participate in the care of these patients. I have no disclosures. The most important thing is that we make the correct diagnosis. Clinical suspicion is based on several clinical features that we've already looked at. And the important thing is to get an MRI and an LP, get an, if you have an CNS oncologist, then that's probably the best, a neuro-oncologist. Otherwise, oncologists who take care of lymphomas and leukemias are the best friends to have to co-manage these patients. And we definitely have to have a confirmatory biopsy, however much we know that the clinical diagnosis is pretty straightforward if you have a typical case. And sometimes you're tempted to want to treat those patients just based on your clinical diagnosis.
Anita, I'm very sorry to interrupt, but your slides are not moving. We are still on.
Moving?
No, we are still on the
They're moving
title.
for me.
No, there is somebody else is also like Yale is also written, and for me also they are not moving. Rima, are they moving
No.
for you?
It's
No.
not even on the slideshow,
No.
is coming.
Yeah, it's just on the first slide.
Yeah, then it's not working.
I don't know why it's, and my slide is moving. Can I maybe
Okay, so
reshare it?
Yeah, can you stop it and share again?
Yeah.
Maybe
Okay.
that
Maybe if you share your screen rather than this particular one, it might work better.
Can
Because
you
it
go
might be
on the
opening a new.
slideshow now? Yeah.
Yeah. How's it now?
No, it's not going on the slideshow, but can you move forward? Like we are still on the
On
first.
my computer, it's moving on the slideshow.
Mhm.
Can you share
Let's
your
see.
screen? Because I think
Yeah, it
maybe
came.
it's opening a new
It came
window.
once.
It came in between, yeah. Can you just close it and share again all over?
Yeah.
The slideshow, click on the slideshow mode.
It is okay. I am on the slideshow mode now.
No, setting.
Setting.
Yes, ma'am.
That's best setting.
Animation.
Setting on my, you mean on the Zoom setting?
No, ma'am. Slideshow mode.
Yeah.
It's not going on slideshow, no? She's clicking.
I'm clicking on mine, it's going, but I guess it's not going for you guys. So there must be something between.
I think there's an option to share your screen instead of sharing the particular file, and then it will show whatever's at the top of your screen.
Right, yeah, but I don't know why. Let me go back to
Can you
share
just
screen.
close it and come back? That might help.
You mean close my PowerPoint and come back?
Yep, share it
maybe
all over
I
again.
gotta do something here. I guess I should share my desktop and then.
Yeah.
Yeah.
But I closed off my PowerPoint, so let me reopen that and make sure I have it open.
Go to share screen before you open the PowerPoint maybe, and then open the PowerPoint.
I think so. Okay.
Yeah, go to share screen.
Yeah, I'm gonna share screen now, but let me see here. Okay, let's see if it'll open now. If I do desktop and then, okay, does that work?
That's better. Yeah.
Yeah, that's great.
Is this
So-
I
working?
think it's working.
Perfect.
All right, sorry about that.
Okay, go on.
Okay, all right. I guess, thank you for having me participate in the symposium on intraocular lymphoma. As I mentioned, all of us are much better off treating and diagnosing and managing these patients, co-managing these patients with oncologists. And so today's talk, I'll try to briefly at least mention what the oncologists will help us with. The most important thing about CNS lymphoma is to make the correct diagnosis, and clinical suspicion and confirmatory biopsy is very important. Most of these patients, when we see them, they present to us with eye findings. So it's mandatory to get them to see a neurologist, get an MRI of their brain, and possibly a lumbar puncture if the MRI is negative, and have a confirmatory biopsy either from the eye or from the brain. And after this is done, then the treatment depends on whether the brain alone is involved, the eye alone is involved, both brain and eyes are involved, or is this a recurrence of something that was already previously treated? And the most important thing after we do treat these patients is to monitor them and have a surveillance to watch for any recurrences or effects in another location, either brain or the eye, depending on where their primary tumor was. So as I mentioned, it's important to get an MRI with contrast, a lumbar puncture if necessary. If the MRI is negative, only then a lumbar puncture is probably needed. And to make the clinical suspicion based on the clinical features, and to do a vitreous biopsy along with a retinal biopsy if retina is involved, and arrive at the diagnosis. I've never used IL-10 levels, but I know Dr. Pulido uses them a lot, and I think other people use IL-10 levels from the anterior chamber to see if you can suspect lymphoma in a patient that has maybe vitreous cells and no other features. They look for IL-10 levels, and these numbers are considered to be sort of suggestive of intraocular lymphoma, 30 micrograms per ml in the aqueous and 65 in the vitreous. And all of us know this clinical appearance. We've seen enough examples. Once you have this appearance, this is very typical sub-RPE location. It's straightforward for us retina surgeons to go do a vitreous biopsy along with a retinal biopsy. And I initially, when I first started, I did only vitreous biopsies, and often the report came back as reactive or indeterminate. So once I realized that retinal biopsy is far more specific in making the diagnosis, I do choose a site that's easy accessible, maybe superiorly, and get a good sample of the retina and laser the edges of the retina and send off for pathology. You have to, the most important thing is to tell the pathologist that the sample is coming, have one of them be in the operating room with you so that they can pick up the sample and not lose it. And there are a few tips during vitrectomy that I consider. One of them is to use your MVR blade and make 2 cuts in a sort of an angular fashion, and then use a curved scissors to cut the remaining part of it. And now that we all use 25 gauge cannulas with valves in them, you have to remember to take the conjunctiva down on that side where you want to put your scissors and get your biopsy, and have a very wide, a fairly large incision. Maybe use a 20 gauge MVR blade and widen it just before you pick up the piece with a forceps that doesn't crush the tissue. So you have something like an ILM forceps that has a little space in the jaws that you can pick up. And then actually, once the retina has lymphoma in it, it's pretty thick. Once you cut it, it just sits there on the surface of the retina, and you can easily pick it up and send it off for pathology. And if you're going to do a vitreous biopsy, these are situations where you suspect that this patient may have lymphoma. And Dr. Pulido already talked to us about how to send those samples. And if you don't have anything in the eye, or if you can't get an answer from the eye, then you look for the brain. And if you find a lesion in the brain, you get a brain biopsy. And this is what the lymphoma cells look like, blue cells that are pretty homogeneous. And once you have a confirmation, you go for treatment. And the treatment has evolved over time. Initially, when I was a fellow, most of these patients got whole brain radiation, because lymphoma is extremely sensitive to radiotherapy. And unfortunately, those days, people didn't know that if you treat they also included the eye fields when they did brain radiotherapy. And if the patients subsequently developed intraocular lesions, they could not get radiation. And in the late 1990s, at about 98, 97, people in Oregon Health Sciences University kind of came up with an management where you can treat the intraocular tumor when patients had already had radiation therapy and they couldn't get repeat radiation for eye involvement. And I'll tell you that in the next slide. At the moment, we have whole brain radiation, systemic chemotherapy, intrathecal chemotherapy, and intravitreal methotrexate and rituximab as all modalities to treat these patients. Whole brain radiation is sort of not picked as the initial treatment now, given that the recurrence rates are a little bit higher with that, and systemic chemotherapy with or without intrathecal chemotherapy, if it's just in the brain, and intravitreal methotrexate in addition to systemic chemotherapy is the treatment of choice. And due to the side effects of X-ray radiation and inability to use it again, if the eye got involved, patients in OHSU began using intravitreal methotrexate, 400 micrograms and 0.1 ml. I think when they first started, these patients were getting it only for vitreous lymphoma. They did not try it for retinal lymphomas. And the dose that was used was 400 micrograms and 0.1 ml twice a week for the first month, followed by once a week for the next month or two. And based on the response, they would hold off on further treatment. Now, intravitreal methotrexate is used as a primary treatment. If the eye alone is involved, it can be used in adjuvant therapy along with systemic chemotherapy when both the eye and brain are involved, and of course can also be used as salvage therapy if the patient develops recurrence of the tumor. The combined therapy is high dose, involves high dose methotrexate and intravitreal methotrexate. This is generally the dose for high-dose methotrexate that's used for systemic chemotherapy, 8 grams per over 6-hour cycle, 6 grams per meter square over 6-hour cycle in smaller patients, or in some of these patients after the initial high dose that was used. I'm not absolutely certain how many cycles they receive. I think they receive about 6 cycles initially, and then based on how they do, they may need further treatment. It can always be repeated. Systemic chemotherapy, sometimes with methotrexate, cytarabine, and hydrocortisone is also used, but high-dose methotrexate is the primary agent playing a role in treating lymphomas. As I mentioned, intravitreal methotrexate, in addition to or by itself, can be used based on where the location of the tumor is. And if you're doing it intravitreally, 400 micrograms in 0.1 ml 2 times a week for the first 4 weeks, then once a week for the next 1 to 2 months, and then as needed. At the moment, whole brain radiation is only used as a salvage treatment. Systemic treatment is the drug treatment of choice initially, and if the patient has a brain recurrence, they can get radiation. The tumor is quite sensitive to radiation and can disappear. If the patient has recurrences, say they've had the systemic chemotherapy and the intravitreal chemotherapy and they have recurrence, based on where the recurrence is, you target the treatment. If it's in the brain, you could do radiation. If it's in the eye, you can do intravitreal methotrexate. Here's an example of a patient that I first saw when I was a very new attending. This lady came to us came for cataract surgery, and we found she had these clumps of vitreous cells along with some lesions in the retina. And I sent her off to the oncologist saying that, please radiate her. At that time, we had not used intravitreal methotrexate, and nothing was done. And then she comes back 3 weeks later and her eye is worse. She's now light perception in one eye and no light perception in the other eye, and the tumors obviously spread around. And when I called the oncologist and said, how come you didn't radiate her? And she came back and they told me that, she'd already had radiation of her there at the time of her brain radiation, because we already knew she had the CNS lymphoma. So at this time, nobody there was no evidence that people had used it for subretinal or retinal tumors. And we injected her with methotrexate, and this was her tumor just about 3 to 4 weeks later. All the lesions disappeared. And I think prior to this, the people in Oregon had used it mainly for vitreous lesions and not really for retinal lesions. And this was the first time that at least I had an experience of using it for retinal lesions. Here's another example of a rare thing that can happen. These patients I did a diagnostic vitrectomy on this man for the first time. It was just a diagnostic vitrectomy. The answer was negative. They told me it was reactive lymphoma cells, reactive lymphocytes. And then I took him back and I did a retinal biopsy. And when I did a retinal biopsy, it came back as lymphoma. He also had second eye involvement. In fact, we got this biopsy from the second eye, from the fellow eye. And I realized by the time we got the final diagnosis, about 3 months, on all the lesions in the right eye without any treatment had disappeared. So they had all healed up. Sometimes you can see the spontaneous resolution after a vitrectomy in patients with lymphomas. So don't be surprised if that happens and think that this is not lymphoma. So that's a pearl to remember. So after you treated these patients either with systemic or intravitreal or a combination of both of them, monitoring is very important. IL-10 levels can be done, and the dose that's considered to be suspicious of recurrence is 2.5 picograms per ml. Clinical exam is very important, and sometimes these lesions don't show up as big lesions because they've already been treated or at least gotten partially treated. So wide field fundus autofluorescence is useful. It'll tell you if new lesions appear, because the RP, these lesions are somewhere in the vicinity of the RPE, and the RPE can get irritated, and you can see them as hyper or hypoautofluorescent lesions. Of course, MRI surveillance is important. So it's a kind of a combination management with oncologists. So in summary, I think confirmatory and correct diagnosis is important to locate the lesion, whether it's just in the brain or in the eye or in both locations, is essential. You select the appropriate plan based on the initial findings. Consider salvage and adjuvant therapy when you need. And so ongoing surveillance is important. In fact, the patient I just showed you, he lived all the way from 2003 when he first presented to 2016. So he lived about almost 13 years after the occurrence of lymphoma, and about 14 years after the diagnosis was made. It took 2 years to make the diagnosis, because the first year he was just followed by his primary care ophthalmologist. And eventually, after 3 vitrectomies and 2 retinal biopsies, we got the diagnosis. Thank you. I have not, I've also used rituximab. Rituximab can be used intravitreally, but the response with methotrexate is better than with rituximab. And if it doesn't respond to methotrexate, you could use intravitreal rituximab in addition as a treatment.
Thank you. Thank you, Anita. And with this, now we come to our session on controversies. I think the first controversy which we have had two talks saying two different things. Like Jose said, it's very silly to do biopsy because you can get most of the information by just taking the vitreous out. And followed by Anita's talk, which said that retinal biopsy is important. And if we look at the sequence of event, we saw Anita showing us that these lymphoma cells are in the subretinal space. David Saraf showing us that only 7% of the patients had actually the cells in the retina. The most of them were either subretinal or sub-RPE. So this is my question to all the panelists before I come to Mark D. Smith, who would like to share his experience. Is it retinal biopsy or vitreous biopsy, or is it one step after the other? So we start with our pathologist friends, that is Sarah and of course Anita, and then we move on to the clinicians. Yes, Sarah.
So my recommendation would be that, and this is what I say when I'm speaking to vitreoretinal surgeons, is then when they perform the vitrectomy, I would recommend a thorough vitrectomy like Jose suggested. And I wouldn't do sort of like an anterior vitrectomy and just hope that there's some cells in there. I'd go as close as possible to the actual retinal lesions. And I think that would be the first step for me. And then if there is no definitive diagnosis achieved, then in competent hands, I think a chorioretinal or a subretinal aspirate could be considered, plus or minus a chorioretinal biopsy. However, obviously it's clear that, such a chorioretinal biopsy could be associated with some morbidity. I just want to emphasize that, it's really important to send these samples to a pathologist who has experience with these samples. I don't think there is any point sending a vitrectomy sample with a question of vitreoretinal lymphoma to a general pathologist who sees these samples perhaps once a year. You really need to have somebody who sees them on a regular basis, at least once a month, if not more frequently. So I think you have to choose your pathologist when you're sending off these samples and make sure that they know that these samples are on their way.
Anita Chan, this is for you. Besides morbidity associated with the procedure itself, chorioretinal biopsy, Jose did mention about CD29 cells above the Brooks membrane. And if you do the biopsy, you are kind of confusing the choroid, which is not touched so far. So does that bother you?
No. So when I trained doing ophthalmic pathology, I trained with Nasty now at Doheny, and he always mentioned to do a chorioretinal biopsy. And the reason was not because the reason was because we wanted to have that full thickness appearance, so we could actually look for the retina, the subretinal, and a bit of the choroid. As if it's a good pathologist, and if you have that tissue, we can actually differentiate between the T cells and the CD20 cells. It's not really an issue for the experienced hematopathologist. And then whether we mix up the tissues and everything, it's not really important because we can tell the difference. And the whole point when we tell, so it really varies from the pathologist point of view when we tell the surgeons, because different surgeons will view chorioretinal biopsies in different light. Some people will just end up giving us a superficial retina. Some people will give us a subretinal biopsy, and some will give us some of varying degrees of tissue, you see. So as a pathologist, sometimes we ask for the full thickness so we can sort of see where it is. Having said that, retinal biopsy is fine as long as you've got the tissue. Sometimes what happens is that it's too necrotic and there's no viable tumor cells left, and it just stains the background of necrotic CD20 cells. So we can say, yes, there's a background of CD20 cells, and it's most likely necrotic lymphoma cells, but there are no viable. So sometimes you want a little bit thicker because they want to see the viable tumor cells, which may be sub-RPE. And it really depends. If you look at the
So, David, how would you do it in UCLA? What would be your protocol?
I mean, I don't know that I'm in a position to answer that question. Colin McCannell is probably the person who has most experience at Stein, and it's beyond my comfort zone. But I would really just defer to the others on this topic. But just because we don't see cells in the retina on OCT doesn't mean that we're necessarily going to get a negative biopsy. There may be cells there that can be picked up. Over 50% of our cases on OCT had subretinal involvement, so I would assume that those would be picked up with a retinal biopsy. I guess the cases where you have just exclusively sub-RPE infiltrates, that probably would be a situation where we'd have to do an investigation to see what the yield would be in those kinds of circumstances with exclusively a retinal biopsy.
So, Mark, you have to tell us something. You wanted to share a case?
Well, I can show you the case, but I can maybe also tell you my own experience with regards to subretinal infiltrates and subretinal and sub-RPE. We're talking about lymphoma cells. So these are usually cells that are, they're not fixed into tissue. And I think what Sarah was saying as a first approach, just trying to flush out the cells from the subretinal space is a good approach. My approach has always been, if there is something under the retina, the vitreous was negative. When I go back, I first try to inject a little bit of BSS, re-aspirate this, and I want to get one of the technicians in the room so that they can tell me whether the sample is in fact cellular or not. And if it is and the cells look good, that's where I stop, if there are enough cells present. If that doesn't work, then I'll go on to a biopsy because I think it's a lot more involved. And what you need here is to be able to identify the tumor, whether we have necrosis, whether there are positive T cells. By the way, they're CD8 positive in the choroid, usually along the Brooks membrane. It really is secondary. Once you identify the lymphoma cells, you've identified the pathology. If you allow me, yesterday we weren't quite sure that we had something from Jose, so Vishali asked me very quickly to present a case. So give me 5 minutes and maybe we can continue the controversy after this. And I think it summarizes a lot of what we've been talking about. If I can get this going now. Do you see my screen?
We do.
Okay, good. So this is 30 years ago when I was at the NIH, when you had a suspicion lesion, we would do brain imaging and we'd do systemic testing, and then we'd do repeat CSF taps. I can remember doing up to 8 taps in order to try and identify lymphomatous cells. The brain biopsy was seen as being definitive. Vitreous was okay, but you still went to brain. I think today, if we have a positive biopsy, it diagnoses the CNS lymphoma, there can be some debate, but if it's a very typical large B-cell lymphoma with some other features, I think we can accept that it is involving the eye. And if we see brain lesions, it might not be necessary to go on to a brain biopsy. So this was a 71-year-old man. He has decreased vision, sudden onset 3 months ago. He's referred as being an unresolved vitreous hemorrhage. I did not see those cluster of cells that were described earlier. We did a vitrectomy. And as these vitrectomies go, they tend to be later in the day. So being able to send a sample to pathology is rather difficult. I think these are samples that need to be sent early in the day. Pathology has to be informed. I usually have my technician take the sample, bring it directly to the pathology lab, which means that he has to take his motorcycle and go over to the center where it can be done. During the vitrectomy, there's a salt and pepper appearance. Vitreous haze is quite high. You'll notice in the post-op picture at 1 week, it is probably typical of a lymphoma. There are also deposits on the surface of the retina. He's treated with some steroids topically, and you can see that a month later, it has largely resolved. So if we are now thinking of a lymphoma, we're going to have to wait for the reaccumulation of cells or try to find other lesions. 2 months after, he gets a fluorescein. I think it's very typical on the right-hand side. The OCT also shows you these changes that are at the level of the RPE with cells either within the subretinal space or just under the RPE. We do a vitrectomy a little bit later, the following week. The cytokine analysis is positive. I usually look at a ratio of IL-10 to IL-6 because IL-6 can be, and IL-10 can both be high. If IL-6 is higher, that usually indicates an infection or just an inflammatory response. MyD88 was positive, cytopathology was positive. I like seeing cytopathology also. The brain is negative at this point. The ocular lesion was treated with methotrexate. As an aside, when this was first developed at the NIH, before it went off to Oregon, we decided to look at 100 micrograms per milliliter vitreous volume, which means that if you have a large eye, you should be injecting more. And I've always injected a combination with other drugs, dexamethasone in particular, phosphate, because most lymphomas respond well to steroids. He was given 7 injections, and then these were stopped because the hematologist, when he finally decided to treat systemically, said it wasn't necessary to treat the eye anymore. And he probably had enough, but the protocol would have called for 2 or 3 more injections. You can see the evolution in the eye. There's a large lesion here to the right, which grew a little bit after the first treatment, but very rapidly disappeared with a good response. And systemically, the MRI was essentially showed small lesions when the ocular diagnosis was made. The CSF, interestingly, as far as I'm concerned, was positive. They considered that it was not, although the flow cytometry probably was negative, but a MyD88 was positive in a few cells. So an MRI was done 3 months later. It shows subcortical lesions in the frontal gyrus. Still nothing is done because there was maybe suspicion of stroke. A month later, there's an increase in the size of the lesion. He finally gets a biopsy, and it shows you that it is CD30 negative, P53 positive, BCL2 MYC rearrangement is present, BCL6 rearrangement is present. So, you know, this is a typical lesion. It also is again MyD88 positive. He receives systemic therapy. Now you can see here that this is a lower dosage than what Anita has suggested, and at this dosage I've seen recurrences inside the eye. He was given systemically rituximab and procarbamazine. Problem with these dosages in an elderly patient is that they can develop systemic side effects, and that's what happened with his kidneys. So was the brain biopsy necessary? Well, the diagnosis, in my opinion, was already established with the eye. But having a delay in terms of doing the systemic treatment avoids side effects. And in fact, we do have some time. He's responded very well to the treatment that was given. Maybe in this case, because this was a secondary vitrectomy, there weren't that many markers that could be obtained. And in the future, maybe it's very useful to have these markers because it has an ability to prognosticate the response. And is a vitreous biopsy necessary if the brain is positive? If you have typical findings in the eye, I don't think so. But if the vitreous was negative and you needed to do, you could always look at the other eye. You could certainly wait and repeat the vitreous biopsy, in this case, just an aspiration. Avoid using steroids at least for a month. And as I've just mentioned, in most cases, just a subretinal flush is sufficient with aspiration so that we don't need to go over to a biopsy.
Thank you, Mark. I agree with you. Like subretinal flush, just a 40G cannula aspirating the cells from the subretinal space, they do help getting a good yield. Is Deborah around? Yes, Deborah, I have a question for you. Anita beautifully showed us the role of intravitreal methotrexate, and there are quite a couple of questions about rituximab. So what we have from the Mayo protocol is you give a combination of methotrexate with rituximab and keep on looking at the IL-10 levels from paracentesis. So what is the preferred treatment according to you, and what is the end point of treatment for ocular lesions?
So I'm going to address the second question first, which is what's the endpoint? And I don't think we have an answer to that. So when I talk to my patients, for me, it's obvious that we need to treat anything that's visual threatening. So lesions in the retina, subretina, anywhere that's responsible for vision. The data that we don't have yet is it necessary to get rid of every vitreous cell? If the vitreous floaters are driving the patient crazy, that's one reason to treat. But we don't as yet have data that would change the patient's long-term mortality by eliminating every vitreous cell. So I don't look to eliminate every vitreous cell. I will be concerned if we see new retinal lesions that I have treatment failure, new subretinal lesions. And I think that's the data that we're missing. Certainly if a patient is scheduled for a stem cell transplant, in those cases, oncology wants every cell gone from the eye. But if we're only treating with chemotherapy or radiation, whether the chemotherapy is systemic or intravitreal, I don't think we know what the endpoint is. And I do not treat to get rid of every vitreous cell. But if anybody comes up with data that says your patient has, you know, a higher percentage of living longer if we get rid of every vitreous cell, I would change my protocol. The other thing is I tend not to use the standard protocol of intravitreal methotrexate, again, because I don't have any data that I need to treat to get rid of every single cell, and it's onerous for the patient. Twice a week for a month, once a week for a month, once a month for a year, intravitreal methotrexate is not nice for the cornea, nor for the patient who has to come in for that. So I work very closely with ocular oncology at Northwestern, with neuro-oncology at Northwestern, and any patient with VRL, and I use VRL, not PVRL, because as we know, sometimes these are secondary. If they have bilateral disease, or even if they have unilateral disease, but that's a little more debatable, we will go to systemic chemotherapy, even if the brain is not yet involved. It's hard for me to understand how a patient can have bilateral vitreoretinal lymphoma without a risk of it being in the brain. These are not two independent organs that are being involved. So we treat all of those patients with systemic chemotherapy. And then I only supplement intravitreally, usually with methotrexate as my first line, if I have disease that has recurred after chemo or is not responding to chemo. So I think I do a little different protocol than most people.
So Anita, you have a comment?
Yeah, I mean, the protocol I put there doesn't mean that I follow that, but that's sort of what it's, this one, it all depends on, of course, how they respond. And I'll tell you, historically, when I first injected in 1999, the fellow that we had at that time had just come back from Oregon, and he was the one who told me about it. And we were shocked within like 3 weeks with methotrexate, she responded. The bottom line is, I think initially it was all whole brain radiation, and then chemotherapy came about, then intravitreal methotrexate came about. I think if you have just the eye involvement, and the man that I showed you who lived for 13 plus years, he only had eye involvement. And it took us almost, from the time of his onset to the final diagnosis, at least close to 2 years. And he never had anything in his brain. He got high dose intravitreal methotrexate, systemic chemotherapy, did not get any intravitreal chemotherapy at that time. And he did really well. For about 5 years later, he developed CNS lesion. He was on constant surveillance by the oncologist, the neuro-oncologist, and by me. And then he was treated again with high-dose intravitreal high-dose systemic chemotherapy and intravitreal chemotherapy because he had a few cells. And he lived for 13 years. He's the longest living guy that I know of. Most literature will tell you 2 to 4 years, they're all dead and gone. But they can do well. And he was about 48 years old when he was diagnosed with it. And so he lived till he was 60. And it's important to know that not all of them die quickly, and systemic, I think chemotherapy is very important, like Deborah said. I don't think when later on there was the shift from the oncologist and I had only eye involvement with no brain involvement, they asked me to just do intravitreal. That's when I did both intravitreal methotrexate and rituximab, but within 3 to 6 months, that lady developed CNS lymphoma. So I think the oncologists had kind of were helping us first with systemic chemo, and we were just doing intravitreal as an adjuvant or in addition. And then they shifted off to, if it's only in the eye, don't do anything systemic and just do eye. And so this lady, I was injecting her bilaterally, and within 3 months of during the time that I was injecting her, she developed CNS lesions. So I think it's gone back now to doing systemic with or without intraocular treatment. And then depending on how they do, you continue treatment as needed. And rituximab per se, as I said, the only time I've used it is in that lady because I used both of them. I was worried about using just methotrexate since they asked me, the oncologist refused to give her systemic chemo, and they told me just treat her for her eye. And at that time, I gave her both injections. And rituximab, I think, is very specific for CD20s. Methotrexate probably has a little bit more wider effect. So I think methotrexate is considered as first line if you're doing intravitreal, and then rituximab only if methotrexate doesn't work or they get lesions in spite of getting methotrexate. And for the biopsy, I wanted to say something because I think I kind of evolved my own sort of biopsy with that first guy that I did. He had bilateral lesions, and the most important thing is to have the pathologist in the room with you so that they can take the sample. And it doesn't matter if it's sub-RP, it's very easy. You just take the MVR blade, puncture into the bulgy lesion. You will go into the subretinal sub-RP space. And this is what Janet Davis taught me, was to put a soft tip cannula on a syringe and suck all that material. When you look at it clinically, it looks yellow because the lesion is under the pigment epithelium. But the minute you make a little cap, open that RPE and put a thing and suck it out, it's all white fluid. As soon as I saw that, I was surprised. I was like, wow, it was yellow and I was all white. So it's really white. The lymphoma cells are white and the pigment epithelium makes it look orange. And it's very easy to get the sub-RPE lesions. And I promise you, there's lymphoma cells, even if it's sub-RPE, they're in the retina. So even if you cut a piece of the retina and send it off, you'll get enough lymphoma cells in that. They're kind of all over. Even if you don't clinically see that they infiltrated the retina, they're there. And I think it's a definitive diagnosis. MIDI88, at least in the United States, not all labs do MIDI88. Mayo Clinic does it, but they won't take a sample from anywhere else. Arup Diagnostics says they'll only do it for blood and not for the eye. So MIDI88 is great in a center like Mayo, where they will actually do it for their own patients, but it's not easy for people who are in other places. So if you're going to do MIDI88, you have to make sure that the lab will accept the sample, and we've had trouble getting samples accepted by any of the outside labs. So I think their pathology is important, and of course you have to send it to an ocular pathologist or a leukemia lymphoma pathologist, and not to a general pathologist.
Thank you, Anita. While we are at it, before we move on to Sarah and Mark, David, I have a question for you. You showed us beautiful imaging biomarkers. What are the biomarkers you are looking for in a patient where you are suspecting a recurrence? Is there anything that OCT will show you before you clinically see subretinal deposits? You are muted.
That's another interesting question that we didn't necessarily look at in the series, recurrent vitreoretinal lymphoma, but I'm not certain. Just looking at some of the cases that were shown today, there were again the characteristic lesions in the sub-RPE compartment that I think probably represent the earliest manifestation. So I think that's where the focus of attention would be with an OCT always, is the RPE and the subretinal spaces with initial disease or with recurrent disease. And it tends to be more advanced when you see lesions in the retina.
Sarah, yeah, go on.
I'm sorry. I think that it's really helpful to get wide field autofluorescence on these patients, because that'll often tell you where to look clinically or with an OCT. So if you see any changes on the wide field FAF, then you can go and do a OCT in that area. So I'll routinely get wide field fundus autofluorescence on these patients as I'm following them.
Thank you. Sarah, I have two questions for you. Liquid biopsy. Where are we with the liquid biopsy? And the second question that Ube asked is there any role of bone marrow transplant? Because we did hear that the cells are coming from outside and homing into the eye. So is there any role?
So perhaps when you say liquid biopsy, I mean, essentially by doing a vitrectomy, that's a liquid
Blood.
biopsy.
No, from the blood. I meant from the blood.
Yeah, so I mean, just to clarify that, I mean, I think liquid biopsy is used sort of, to mean blood biopsy, but it's, okay, sorry, I'm being pedantic. So I don't think we're at all far with analysis of blood biopsies in the case of vitreoretinal lymphoma, because I think we don't really 100% know what we're looking for within the circulating cells. That is in contrast to, say, metastatic carcinoma, lung carcinoma, which is typically used to look for circulating carcinoma cells. But yes, there are these suggestions, and I think it was first raised by Andrew Dick's group, that the neoplastic cells are actually forming within the bone marrow, and then they home to the immune privileged sites, and in the case of vitreoretinal lymphoma, then to the subretinal space. So I don't know how solid that evidence is, to be honest. And I think it was Deborah who said that, if you've got bilateral disease, then surely there must be CNS disease. We actually, many years ago, I think it was prior to 2000, we had a case of a 46-year-old man who had a presentation in the eye, and we could demonstrate it was a B-cell clone, and we did the sequencing, and then when he developed a recurrence in the brain and the opposite eye, we could demonstrate that all of those B-cell manifestations had arisen from the exact B-cell clone. So it wasn't as if there was something perhaps arising within the bone marrow and then spreading. It would have appeared that it manifested in one eye and then had home somehow to the brain and to the opposite eye without significant evolution of that clone, and that they were all the same manifestation of that B-cell clone. Which brings me to a point that I'd like to emphasize with respect to CD20 and rituximab. It's been demonstrated with systemic diffuse large cell B-cell lymphomas. If patients are treated with rituximab or R-CHOP, for example, that with time you can get the development of relapses because the B-cell clone has evolved and it has lost its expression of CD20. So I think when we're assessing vitreoretinal lymphoma patients, we always have to keep that in mind, that the clone will continue evolving, and that perhaps the rituximab treatment is no longer going to be effective because the B cells, the neoplastic B cells, no longer express CD20 on their cell membrane. So if you have a patient where there's a suspected recurrence, then the pathologist should consider not only CD20, but perhaps another B cell marker such as CD79A, because in the meantime the B cells may have lost their expression of the CD20 and therefore be no longer sensitive to rituximab. So I guess that they're just the 2 points that I'd like to make, and I'm not sure whether I've answered both of your questions now.
But that's a good point, and that brings to the last point of discussion about multimodal therapy. Like, we are the only ones in the ophthalmology who are treating with monotherapy a malignancy, because most of the other systemic branches is all about combining the therapies. So Mark, there is a question for you. I know you combine methotrexate with dexamethasone. So what is your take on that?
Well, you've just already summarized my take on it. From the very beginning, I've never been in favor of simple monotherapy. The very first patient that was treated was methotrexate and thiotepa. Problem with the thiotepa is I saw a pressure increase in the eye and I got concerned about it, but I also used dexamethasone. So no, I never used the monotherapy. The second thing about methotrexate is the twice a week original treatment was because I had no idea of what the half-life of the drug was inside the eye, and it turns out to be about 72 hours. So once a week is perfectly okay. And we can give it once a week for 4 weeks. And again, my concern is 100 micrograms per milliliter was based on what was given intrathecally to the brain. And so if you think of an eye that can be hyperopic at 3 milliliters or myopic at 10 milliliters as an intravitreal space, giving a fixed dose of 400 microliters makes no sense. And I think some of the occurrences that we've seen early with methotrexate may be because we were underdosing. And on the other hand, if you have a recurrence, they seem to respond very nicely to methotrexate. So it's a good approach. Rituximab, by the way, in a few cases has been shown today to develop vasculitis. So we have to be careful about that also when we use it. But it may be a very good second alternative if the patient doesn't really respond to a decent dose of methotrexate.
John and Rima, any point that I have missed and you would like to discuss with our panelists?
I think we have covered most of it.
Yeah,
Well,
I think
Yeah, I think-
I would just like to reiterate saying that you don't use monotherapy. I mean, all of these therapies are all used in combination, and I think it's evolved over time. I think from radiation to systemic chemotherapy, then to just monotherapy in the eye, if it's only eye, and now it's back to everything. So other than radiation, which is kept aside because of the side effects and the fact that the response with chemotherapy is much better than radiation, possibly because it affects the whole body. And I think the main line is to start systemic chemotherapy with or without additional intravitreal therapy as needed. And then watch for it. There was a question about toxicity to the cornea. Yes, you could get toxicity to the cornea, but so far I've been lucky enough not to notice that. And, sometimes, I mean, I have never really treated them twice a week because it's always been in combination. I've treated them once a week and treated as needed. I don't think you want to individually treat with only one modality at the moment. I think is systemic with or without intravitreal.
So one question I have, do we get the message that intravitreal methotrexate is primarily the first choice? Because that's what we do. And rituximab is in case methotrexate fails or there is toxicity. So is that the consensus by the panelists?
Yeah, I think so.
Yes.
And one other thing for the audience is to know what methotrexate to use. There's intrathecal, there's methotrexate that's used for systemic therapy, and there's one that's only used for intrathecal. And if you want to use in the eye, you prefer to use the one that's intrathecal. It doesn't have any preservatives. So it's important to look for that on the label on the methotrexate. In fact, now that I'm not at the university where the pharmacy would make my methotrexate and send it to me whenever my patient came and I could pre-order it, I have to make my own methotrexate. So we have methotrexate in our office and we just pull it out and inject it. We also use it for some patients with PVR. So we have it in stock and just use it in a private office. So it's simple to do. And
so there are two questions related to BTK inhibitors systemic therapy. Would you treat them systemic with BTK inhibitors and combine it with intravitreal methotrexate?
I don't have any experience with that, so I can't.
Deborah, you have any experience?
I defer all systemic chemotherapy to our neuro-oncologists. So I have no answer.
Okay.
I think that's the important thing, is that you have to work in combination with the oncology department. Hopefully it's better than my last experience recently where, behind my back, somebody's being told you don't need any intraocular treatment anymore. But, I think that's an exception. It's important that if systemic treatment is necessary, that it be given by people who can follow the side effects also, because they'll develop side effects with this treatment.
But is the consensus still that if the disease is isolated to the eye without CNS, or non-CNS systemic involvement at all with comprehensive investigation, that we only treat the eye? Is that pretty much the standard of care, or we are
That's
treating
not what I do.
systemically?
No,
I treat them systemically, always.
yeah, you have to treat
Even in the absence of any systemic findings? Yeah, that
Yes.
would seem to be the logical thing to do.
I've discussed this at length with some neuro oncologists, and their argument, at least, and this goes back several years now, 15 years about, but this is my recent experience also, is they don't mind us treating the eye. They're concerned that if we start treating systemically with heavy medication, we're exposing the patient to side effects, potentially very serious. And this is more important if the person's old. And it may be better just to wait for the tumor to appear inside the brain and start treating it at that point. And in some ways, if you think about it, for a young person, I would prefer having the treatment both systemic and ocular. But if you're above 70 and you're going to treat systemically with high-dose methotrexate and rituximab, you're going to get side effects. And is that better than waiting a few months and then treating and getting a good response? You know, you're going to lengthen their life for about 5 to 10 years. Maybe you're lucky it's longer, but you know, the average survival is about 5 to 6 years. That hasn't changed over 40 years. So I tend to look at it in terms of age. Younger people, for sure, I push. If they're older, I'll treat the eye and make sure that they're followed very carefully.
Marc, as you've gotten older, has your definition of older changed?
Of course it has. So it's evolving. You can see it has to evolve.
I remember when you had a full head of hair, my friend.
Well, with this, we come to the conclusion of a very, very informative webinar where we learned a lot, all of us, new things, few things we don't have the answer yet, but I guess gradually we will reach there. Thank you very much, everyone, for being with us, for attending. Please fill in your evaluation form for CME credit, and we see you next month, webinar on cystoid macular edema before we go for the summer break. Thank you very much, everyone. Thank you, panelists.
Thank you.
Thank you.
Thanks,
coming.
everyone. Bye-bye.
Thank you.
Bye.
Thank you very much, Vishali. Thank you. Bye.
Right.
Bye, Vishali.
Bye, Anita.
Bye-bye.